In paleontology, we're usually looking at what has fossilised in order to work out what life was like in the past. Sometimes, though, we have to work the other way around to give ourselves some perspective. So today we're going to be looking at what is alive now and thinking about what would fossilise. Here we are again in Aberystwyth, on the coast of Wales, on a rocky shore looking at what is living here. I should say to start with that unfortunately the wind picked up a lot and there wasn't much usable audio, so I'm going to have to do voiceovers for a lot of it. What I'm looking at today is what is alive in this environment and what could fossilise, under the sort of circumstances that we would expect to see in the fossil record. And the first thing to bear in mind is this is a rocky shoreline. These places are areas of erosion. The sea is coming in, it's gradually wearing it away, it's chucking pebbles at it, and as a result, the entire environment is likely to disappear in the long term. This whole place is not a good candidate for being fossilised at all. That said, there are a couple of ways we're going to imagine that it might happen today. So, first of all, let's think about the possibility of maybe a surprising earthquake which drops the sea floor by about a metre or so, and allows it to be covered with sand in one massive storm and then forever undisturbed. So that would eventually become a sandstone on top of this rocky deposit which was the original bedrock. And things might survive as fossils there. The other thing we can possibly consider is if there is maybe just a little hollow offshore in this environment, just beyond the waves there, where the seafloor sort of dips down into a little sheltered part, and during a storm things are swept off, washed away from these rocky areas and deposited in that little hollow, there to be buried by finer sediment, maybe silt or mud, and possibly fossilised for posterity. How would that look? What differences would we see between these two types of fossilisation, and what would that tell us? What would we interpret from that about the ecosystem that was here at the time? And most importantly, what does that tell us about our chances of getting any interpretation of the fossil record actually right? Understanding any of that depends crucially on knowing what is there, including the small stuff, and also what they are made of, and what will happen to those materials over geological time. So it's a multi-layered approach, but critically it's a question of firstly seeing what is there, and acknowledging that it's not just the obvious things, it's everything, which might be slightly harder to find in some cases than in others. Almost everything that you see at first glance is seaweed, and quite a diversity of it. There is quite a lot of brown algae, like this rack of some sort here. There's lots of very delicate green algae, and then there are these sort of pink and white crusts, which we'll come on to later. Algae is actually extremely soft. It's made of cellulose, and in terms of fossilisation potential, it doesn't have much. It does happen. You need exceptional preservation, you need conditions of deposition, usually in anoxic environments where the decay from bacteria is slowed down, but it's unusual. Sometimes the holdfasts, these tough bits, might have a better chance, but in general you're not going to get much in the way of algae preserved at all. So those are all wiped out of the equation straight away. There's an exception though. These pink things here are red algae which also secrete a skeleton of calcium carbonate. So the same material that we have in the shells of clams, mollusks and so on, and although they're very delicate, do give them a chance of being fossilised. Likewise, these structures here, these crusts on the surface, are a lithothamnian type alga, which is again a calcareous alga. It makes a skeleton which then is adhering to the surface of the rock, it's encrusting it. You can't detach it from it, at least not without a penknife or chisel, but that stuff is there and is already theoretically a fossil. It's the sort of thing that is going to get fossilised very easily if you just bury it in sand and leave that environment here. None of that is going to be washed out to our little sheltered hollow offshore though. So in that environment you would never know that this alga was here and so abundant. The green and brown algae, these ones could be ripped up as fragments and washed out, and if the conditions were quiet enough and anoxic enough and undisturbed enough, it's just possible that you would get fragments of those being preserved that would tell you that there was a rich environment full of these seaweeds somewhere nearby. But you would need exceptional preservation. Normally, in most environments of offshore mudstone, you would not find anything resembling algae at all. It just wouldn't preserve. Next on the list of obvious candidates for fossils, we have limpets. These are the common species, but they are abundant on these rocky shores. And of course they have a very tough shell. They are very strongly locked onto the surface by extremely strong muscles, and you wiggle them, they clamp themselves down and you can't move them. If this is buried by a sudden surge of sand, they are going to be fossilized in place. They have a very tough shell, it's very strong, made of calcium carbonate, and that is going to survive. They also leave traces of themselves though. You can see markings on the rock, little circular marks, and these are the scars of their home bases, where limpets come back to night after night, wriggle themselves down and scratch away a little ring where the shell was. So you can see where limpets were, even if the shells are dissolved by acidic pore waters or something like that. For our other scenario, these limpets are not likely to be detached from the rock during a storm. They can cope with that quite happily. But of course, some of them do die, and you end up with loose shells washing around in the sand, and those can be transported out and buried in this deeper environment. You may not get many of them, because as they're washed around on a rocky coast, they eventually get smashed, broken into tiny fragments, and they're not recognisable anymore. There are a few other types of snails here as well, and you'll find them scattered around the high tide mark, or around the tide mark, and well, there's actually a reasonable diversity of them here. These are not nearly as well attached as the limpets are, so you'll expect to find those washed out as shells, but equally, if it was covered in a flood of sand, you'd expect to find some of those turning up as well. Again, an aragonite calcium carbonate skeleton, so their shells, they're likely to appear in the fossil record. Next on the really obvious list are of course barnacles. These are highly modified crustaceans that are adapted to a life encrusting rocks in the intertidal zone. So these are all quite happily alive, despite the fact that the water isn't there anymore. So they're highly adapted, they are solidly cemented onto the surface, and these, if they are buried by sand, because they have again a calcium carbonate skeleton of plates, are going to be fossilized fairly well unless the whole lot is dissolved. So they've got a good chance of being preserved if you dump lots of sand on top of it. However, in terms of picking these up and throwing them out into the sunset, that's a lot less likely. The individual animals are firmly attached and they're not going to come off, but when they die, the skeleton breaks down into a series of plates, and those little plates can indeed be washed around and swept out to sea and buried along with sand. You find pieces of these plates in sand samples if you pick them up from beaches, and so you would get a record of these offshore as well. After that, you have to start looking a little bit harder. So you get the odd fragment of other shells. For example, this one is a common whelk. But you see those holes all over it? That's sponges, that is. Clyonid demosponges, which actually live subtidally, they'll just be offshore, and they bore their way into shells. There are also trails of evidence of snails that have been wandering over the surfaces, but those of course won't fossilise at all. It's surprising how few species make up the great majority of what you see that's actually alive. So now you start looking closer. Turn over the rock, and what do we find? Nothing. At least nothing significant. Bit of dead seaweed. Which is surprising. Again, we're not looking closely yet, but turn over another and we find one snail. So we're going to have to start looking quite a lot closer.
Other animals may be harder to spot, but there's still a few things left that can actually be fossilised.
Look closely at these brown algae and you notice little white tubes stuck to the surface. These are also made of calcium carbonate, and they are made by little annelid worms, spivorbids. There's another video about these. However, there's more to it than that if you look very closely, because there are also patches of interesting looking stuff near the tubes. These are bryozoans, moss animals is their common name. They're colonies of tiny little creatures which have filter-feeding tentacle arrays that you can see if you look really closely at them here. They're actually quite diverse and they're very abundant once you start looking. It's just that the individual animals themselves are tiny. There are fossils of bryozoans, and many of them, but they are ones that have a calcite skeleton. These ones we're seeing here are entirely organic. They will rot away, they will disappear, and they will leave no trace in either of our fossil situations. There are quite a diversity of species though, including some that specialise in growing on the wide flat fronds of seaweeds, others that tangle in the roots, and many of these can be washed up on beaches where you can find pieces of them and have great fun trying to work out what diversity there is and seeing if you can identify any of them. Another encrusting animal is a sponge. This looks like a Hymenyacidon species, very commonly found in the intertidal zone, and they are amorphous colonial species that live attached to rocks in places that don't dry out much. So these do have a skeleton. They have minute silica spicules made of opal, and these will not really fossilise in shallow water. They dissolve very easily in these sort of conditions, and they're so small that they're likely to vanish almost instantly. So these again are likely to leave no fossil record, whether they're buried by sand or have little bits of it torn out, and even the spicules themselves being washed out and buried in mud elsewhere. In this environment, we're just not going to see them.
This is a good one. Honeycomb worms. These are colonial polychaete worms that actually make make tubes out by sticking sand grains together. And they cement them to make fairly solid looking structures which can even build up into reefs. So surely these will be fossilised. Well, if you cover this with a mass of sand, would you actually be able to see it? It's a bit of a challenge because once you cement lots of sand together into a sandstone, how is that different, the tubes?
In terms of breaking off pieces of this and washing it out with the sediment, that can indeed happen. And in the sand around here you do find little fragments of these tubes made of sand grains that are glued together. And if that was deposited not in sand but in something like mud or silt, that would be recognisable. We would be able to find that. And in fact there is a decent fossil record of various worms that stick sediment particles of various types together to make tubes in which they live. So yes, we'd see them where fragments were washed out and buried in mud, for example, but we might not be able to recognise them if they were all here in place, but buried by sand, made of the same stuff. We spent a while looking for sea anemones, and failing before Lucy found one of their relatives. So this, of course, is a jellyfish. It's the common moon jelly, one of the most common species around the UK, and they don't look so good out of water, unfortunately. When they're swimming and you're sort of snorkeling with them, they're amazingly beautiful and elegant creatures with very graceful movements under the water. These washed up ones don't really do them justice. Of course, to preserve or fossilise a jellyfish, it takes really extraordinary conditions. There are a small number of records throughout geological time, mostly of the impressions that they leave in sand, but almost all of the time, nothing would be preserved of this. We'd never know it was here. Exactly the same is true of this, a strawberry anemone lurking right next to where we found the jellyfish. These are, like the jellyfish, totally soft-bodied, but they happen to spend their lifespan attached. In fact, sea anemones and jellyfish actually have a very similar life history, in which they spend some time as a medusa phase, the free-swimming one, and sometimes as a sessile polyp. It's just that in the anemones, the polyp phase dominates their life history, and in the jellyfish, it's the medusa stage. Attached to rocks in crevices and underneath stones, you find yet another of their relatives. These are the tiny, barely centimetre-tall colonies of hydroids, hydrozoans, another of the same group in which they do have a free-swimming medusa-like phase, but most of the time they spend as these little colonial organisms in branching tubular colonies. Although these ones do have this tubular skeleton, it's made of chitin, and chitin is a polysaccharide technically, which means basically that it rots. So again, virtually no fossil record of chitinous hydroids. They turn up very, very rarely, but you need exceptional preservation to find them. That said, there is an organism that we find as fossils occasionally called Plumelina, and this is normally found in Ordovician to Devonian rocks in sandstones. These are thought to have been chitinous hydroids, so it appears that in these larger, more robust ones, sand can at least retain a mould, an impression of these larger colonies, even if they do ultimately rot away. Okay, so everything we've looked at so far, pretty much, has been either permanently attached to the rocks or tending to cling to it, like the snails. But of course, there are lots of much more mobile animals in these places. And for those, we need to actually go looking with a little more effort. So one way of doing it is to use a sieve and just waft it around through the weeds and see what falls out into it. You can look underneath stones carefully. You can brush the furry stuff that grows on the pebbles into a box of water. There's all sorts of ways of finding things. And of course, as you all remember from your childhoods, one thing that you're likely to find very quickly are shrimp. Particularly things like common prawn, which are extremely common in general, but they're fast-moving, they tend to lurk in corners where they're difficult to spot, and they can easily be overlooked at first glance. But they're there, and they're really beautiful when you see them up close. There are other smaller arthropods to turn up with this sort of method as well. So this Lecanosphaera is an isopod, a relative of the familiar terrestrial woodlice. And this was also found clinging to some of the soft green algae. All of these little crustaceans are made of chitin. Crabs and lobsters, for example, can have a little bit of a biomineral involvement with them as well, but mostly, the smaller ones especially, are chitinous. And they, again, do not fossilize. If you buried them in sand, they just decay and disappear. If you swept them out into a quiet, muddy place, then you might have some chance of finding remains of the more robust ones, but normally anything like this is a sign that you've got a degree of exceptional preservation happening. So for 99 point whatever % of the fossil record, you just won't see these at all. So in this thought experiment, we're going to assume that we're just not going to find them. I would really like to have been able to talk about starfish as well, but on that day we didn't find a single one, or sea urchins. And starfish are particularly interesting though, because they have a skeleton made of calcite, which is quite easily fossilizable. Except that there's very little holding that skeleton together. It's made of hundreds or thousands of tiny little plates, which, once the animal dies and starts to rot away, effectively disintegrate. They just separate, wash around in the smallest current, and you're left with almost nothing. This is the case for most of the echinoderms, this group that includes starfish, and in the fossil record, mainly the crinoids. But you can get complete ones, and you don't need soft tissue preservation, as long as things are buried alive or very recently dead, and then not disturbed. So, in our situations here, if you were to have found a starfish and it was covered by sand, then yes, if that was left undisturbed thereafter, you could get a complete starfish fossil. Similarly, if it was picked up by a storm and swept out and buried in a pile of mud, then that also could become an entire starfish fossil if it was then undisturbed. And that's the difficult bit, because you only have to look around to see how much disturbance is going on. Sediment is laid down, but then it's picked up again. Then another storm comes through. It's washed around again and again. You need really unusual conditions to bury something like that intact. And if that doesn't happen, then all you're left with, if you're lucky, are a few of those individual plates, the tiny pieces of the skeleton. If a complete starfish is unlikely, though, then the things we need to look at next are practically impossible. Because all we've been looking at so far are the larger organisms. There are many other things living here which are vital parts of this ecosystem, and which in fact basically construct it. They are the ones that maintain most of what goes on here. And to find those, you need to start looking in a slightly different way. So you can swirl some sediment around in a glass jar, so things in the sediment are borne up by the water, then pour that off, and then look at things like that under a microscope. You can take sediment sample itself and look at that under a microscope. And each of these approaches will show you slightly different things. Here's a very active ciliate, for example, a single-celled protist that's whizzing around looking for anything that can eat. It always amazes me how much behaviour single-celled things like ciliates actually have. And here it is again, coming in from the top right, but in the centre there's a little wiggly thing, which is a nematode worm. This is one of the roundworms. Many of them are parasitic, but some aren't. Just change the lighting over, and so you can see it better. And there it is, just very distinctively sort of wiggling. They are mostly on that scale. Some of the parasitic ones can get a lot bigger. Now there's another little ciliate, a tiny little round blob bumbling around everywhere. They do that sort of thing. Moving on to another worm. This one is not a nematode, but a nemertean. It's a ribbon worm, and this is probably a very young juvenile. It's only a few millimetres long. If it's the one I think it is, which is the bootlace worm, they can get up to about 30 metres, which is slightly ridiculous for one of these things. But there we go, they're actually quite fascinating. You can see it's ingesting lots of just general organic stuff to digest and see what it can get out of it. There might well be a lot of sediment in there as well, but it doesn't really care. It'll just digest what it can and leave the rest. Another little ciliate bumbling around there. And it's quite sort of hypnotic when you zoom in closely and see the workings of these transparent animals of this size. There you go, that's quite a mouthful, isn't it? Yeah. Open wide. This one should be more familiar, but can you see it yet? Let's go in a little bit closer and closer again, and oh yeah, there it is. There is a snail. This has got to be one of the earliest growth stages of, well, an unidentifiable at this stage little snail. You can see it's just got one little whorl, which is a good indication that it's very immature. Later on it'll become visible as to what it actually is. Snails, of course, would be potentially fossilizable no matter how delicate they are. And here's another one that fits that. It's not a mollusk, but it does have a shell. And we'll just find it in a moment, I think. Here it is. Oh yes, it's actually rather an ornate shell. It's got lots of little dimples all over it. It's got a hinge line along the top. It's really rather pretty. This is an ostracod, a seed shrimp. They are tiny little crustaceans that are mostly within the shell, but in this case only an empty shell. The animal, alas, is no longer with us. Some of these critters are much more elusive though. This one you can see there's something moving around in the murk, but you'll have to take my word for it because I did get a brief glimpse of it. But it's a harpacticoid copepod, a ubiquitous little group of tiny little crustaceans that are best known probably from the genus Cyclops that you can find in pond water all over the UK and probably most of the world as well. But it's there, it's another part of the ecosystem that would otherwise be totally overlooked, and they're probably very abundant in the finer sand and mud. Okay, so those are things that we can find alive just living in the sediment. But we can also just go to the beach and pick up some of the sand there and see what has made it into the sediment as a sort of a proto-fossil. And the best way to do that is just to choose a bit of sand, put it in a little box, and have a look at it under a microscope and see what's in there. Now the sand itself is quite interesting and lots of diverse crystals and so on, but there are obviously bits of organic material here. You zoom in on this and you see it's a little stick-like thing with holes on the side. Zoom in further and you see the pores on it. This is a bryozoan, a Chrysia species. They're very common attached to other bryozoans and to seaweeds, but it's really nice to see. There are also lots of shell fragments and bits of barnacle, and even some broken but semi-complete and recognisable shells. This is one among diverse little gastropods that are micromorphic. They don't get bigger than a few millimetres. This is a full-grown one. Unsurprisingly, most people don't even know these exist. There are also tiny oyster-like shells that you can find attached to seaweed washed up on the shore. Some shells are more mysterious though, like this sand-sized little white thing that almost looks like a bunch of bananas. This is a miliolid foraminiferan, a single-celled amoeba that secretes a shell of calcium carbonate that can be fossilised. Some of the shell fragments that you find as you're scanning around might almost seem recognisable, and others almost certainly will be. And the more time you spend looking through sand, or just on the beach generally, or in rock pools, the more things you will find. Having a survey like this, just taken place in a few hours, is not nearly long enough to find the full diversity of what is there. What it does tell us is what the most abundant things are. And when you're comparing with the fossil record, we have to assume that most of the time what we're going to be seeing is only the most abundant things, because the chances of any one individual being fossilised are so incredibly low that most of the time we're only going to be seeing the most abundant organisms in an assemblage. And given that, we can now compare directly with what we expect to be fossilised in a site like this, and compare that with what we know is living here. So let's just see how bad an approximation to the real world the fossil record is likely to be. So what I've done is put together a table that tries to summarise roughly what we would expect to find fossilised and what we wouldn't. So the first thing to notice is that all those things we pointed out at the very beginning, those various groups of algae, are not going to fossilise at all in terms of the soft ones. Calcareous ones, in the area where it is buried by sand, where they're actually preserved in situ, then probably. Bits that are swept out, only fragments of the corallina. The others you would not know about. Then you've got the shells. So a lot of these, again, if they're buried in situ, they're quite likely to turn up. So you've got various gastropods there, grey top shells, limpets and so on. And some of those at least will be washed out fairly intact and may even be preserved complete. But a lot of the ones like the limpets are going to be damaged because they'll only be the empty shells, which will have been washed around for a while and broken up and then finally deposited in the sand. After that, the picture is a lot worse. So we have things like the honeycomb worms, fragments, yes. The bryozoans and the sponges and the sea anemones, not a hope. You might have noticed that I've not mentioned seabirds at all. There's a reason for this. Although seabirds are actually very common there, that's only from a perspective of a much larger scale. They move around a lot. So when you're dealing with an area of a few square meters of seashore, you're highly unlikely to encounter a seabird in those few square meters, especially a dead one. As we walked around, there were hardly any remains of seabirds, just a few bones here and there of fleshly dead ones, effectively, ones that have been scavenged but not yet broken up by the waves. And their skeletons are actually extremely fragile. The bones are partly calcium phosphate, but they're also mostly organic, and they do break down very easily. When they get washed around by the waves, they are smashed into pieces. There might be tiny fragments of bone to be found, but in the sand sample I didn't even find any of those. So although they are common on a sort of human landscape type of view, in terms of what would make it into a little bit of the sediment as part of the fossil record, they're basically invisible. The second half is even worse, if anything. So here we get into all the very soft things and the chitinous creatures. So we've got things like the jellyfish and the hydroids. The hydroids, I guess, we did decide technically some of the largest ones might be preservable in the sandstone based on what we do see in the fossil record. But they're actually extremely rare fossils, even in sandstones, and those are the large ones. So that the small ones, almost no chance. The common prawn I've given a question mark, possibly in the mudstone. If you get a little bit of slightly exceptional preservation, there may be chunky enough that at least the carapace might be preservable, but you're really going out of the realm of normal fossilization at that point. The other little arthropods, the nemertians, the ciliates, yeah, right, and not a chance at all. The only things left on here that do have possibilities really are the oysters and the forams, which both have calcite skeletons. I also have fish on the list here, and this is something I haven't mentioned up till now, but there were a very small number of fish in those rock pools. When I say a very small number, I saw 2 or 3. Basically, I think they were rock gobies, very young, maybe a centimetre or 2 or 3 long, and they were extremely quick. Their skeletons are, of course, far more delicate even than seabirds. So even though they might be buried by the sand, they would not really have any chance of being fossilised. If they were being washed out into this muddy area, again, almost no chance. You might, again with some exceptional preservation, get a complete skeleton being preserved, but you'd have to be lucky. So overall the picture is not exactly wonderful. The impression you would have from this fossil assemblage is in the sandstone you would get a sort of rocky shore fauna of barnacles, limpets and a few shells, maybe the honeycomb worms, and not much else. And so you would think that it was a really harsh environment at first glance, something with almost nothing living there. You wouldn't have an impression of this lush growth of seaweed, for example, and all the things living on that, because both the seaweeds and the things living on them are not going to fossilise. So you wouldn't be able to tell the difference between an assemblage or a community which was basically exposed rocks with virtually nothing on, to what we're seeing in those rock pools. They would look the same in the fossil record. The only hint you've got is that there are these various snails, the top shells and the limpets, which are grazers. So those will be dependent on there being some algae to eat. But you don't know, because they're being washed around a bit, whether the algae were actually there, and you don't know how much there was. With the mudstone, you might get better preservation, but the situation is actually even worse because you only get the mobile things. So things like the lithothamnion, the corallina, you'll only see little fragments of it to let you know that they were there to start with. Most of the shells are going to be damaged in some way. They're not going to give you a picture of the whole ecosystem at all. It's a really limited view of what was living in this area. The moral of this is not that it's impossible and we should all give up. It's rather that we have to really understand what it is that we're looking at, as far as we possibly can. So you have a slab like this with some rather nice shells on it. What do you do with that? Do you assume that what's on here is all that was living there? Of course not. What you do is work out as much as you can from what is preserved, and then start to piece together the clues to what else must have been there in order for this ecosystem to work. And then that's only going to take us so far. So in relatively young rocks, say back to Mesozoic, that's actually a reasonably good basis for understanding how the ecosystems work. In the Paleozoic, and especially the early Paleozoic, it's much more difficult because life was different back then. We can still make some assumptions about different parts of the food web, what was necessary for the survival of one group versus another. But then we are really dependent on those rare cases where we do get most things preserved. These cases are exceptional preservation, where soft-bodied animals, plants, are all fossilised in the same place. And it's those little windows that then give us a perfect sort of springboard, a template, or a sort of Rosetta Stone almost, for interpreting what we see in the rest of the fossil record. And I think that's plenty. This has gone on for quite a long time, but thank you very much for staying to the end, those who did, and I will do something probably a little bit shorter next time. Thank you very much, and goodbye.